Welcome to Lab 6, where we'll be discussing seed viability and germination testing. The first thing that we need to know are all the parts of a seed. So this is a typical bean seed, and we're going to use lots of beans in our experiments this week because they germinate very rapidly and we can see results very quickly. On the outside of the bean is the seed coat, otherwise known as the testa, and then the big giant piece that you typically are used to eating as a bean that's very nutritious is the cotyledon. The cotyledon is the storage tissue that the plant has made from a triploid endosperm and becomes food for the growing plant. The growing plant is made up of three different parts. The long white piece that you see here is called the radicle, and the radicle is eventually going to grow into the root. This curved piece that I've made in orange is called the hypocotyl. The hypocotyl is the curved piece that ends up being the root-shoot axis, so the point at which it switches from being a root to a stem in a plant. And then lastly, you'll see something called the epicotyl. The epicotyl are the first true leaves that will grow out. Usually they aren't green while they're in the seed because they have not been exposed to light, so they're not photosynthesizing, so they'll just be white in color. The first kind of test that we're going to do is called a cut test, and we're going to do this with fava beans. So with a cut test, you're opening up the seed to see if there is an embryo inside. We're going to take a whole pod and pull out different beans from this pod. The area that you see that has like a little scar on it on any given bean is where it was connected to the parental tissue of the pod. And then we're going to cut that open. With the fava beans that we're using, they have a seed coat that's on the outside that we would remove. And then on the inside, you've got these cotyledons. And then we're looking for this little structure on the inside that would have the radicle, hypocotyl, and epicotyl. And if you see that embryo on the inside of your seed, then you know that it was a viable seed. At least it was until you cut it open. Now it's not viable anymore after you've cut it open. Then we're going to do something called a float test. And a float test just determines if the seed is more or less dense than water. So we're going to take a big Tupperware container. We're going to throw 100 seeds in there. The ones that sink we will consider viable, and the ones that float we will consider non-viable. We're going to go one step further with this experiment and then plant out those seeds to see how reliable our float test ended up being. So this will end up being a direct measure of seed viability. Lastly, we're going to do something called a direct germination test, and for this we're going to use both mung beans and black beans. First, what you want to do is you want to take your beans and you want to soak them for 30 minutes. You're going to need 20 beans for each experiment, and there are 3 different treatments, so you want 60 of each kind of bean. And then after they've soaked for 30 minutes, we're going to put them in 20 of them in just water, 20 of them in a moist coffee filter, and 20 of them in a bag of soil. And we will check them daily to see how many have germinated and how long it takes for all 20 beans to germinate for each different type.